Hello. We continue chapter 21, the lymphatic and immune system, and we are starting the immune system, part 1. Here's your table of contents. And first of all, let's talk about immunity and pathogens. Immunity is an ability of our body to resist damage from foreign substances as pathogens. So what are pathogens? Those are any agents causing disease, but it's not exactly correct. Usually we are talking about living microorganisms that are capable of producing infection, causing disease. Poisons are not included, or some factors like scalding your hand with hot water. Hot water would not be a pathogen. So mostly we are talking about cells and living microorganisms. Okay, types of those microorganisms. Let's start from cancer cells. They are our own living cells that start reproducing too fast and turn into cancers. And our immune system usually can protect us against them. Next one, some harmful chemicals. Here's your example. Prions, or some people pronounce it prions. Those proteins have very complex structure. They are non-living because they are not complete cells. They cannot reproduce by their own, but when they get into living body, human or animals, they can cause disease. Example would be mad cow disease. In humans, it can affect the brain. Next one, microorganisms. Those would be bacteria, viruses, fungi, protozoa, parasites, specific proteins we just said about them. Okay, viruses. Here is a virus. Well, virus is living and non-living at the same time. Generally, we say if it doesn't have a barrier border like cell membrane, it's non-living. But at the same time, when virus gets into our cells, it can cause disease. It starts reproducing inside of our cells. Examples of viruses would be viruses causing hepatitis, several of them, herpes, warts, chickenpox, measles, tetanus, and a lot of other diseases. Next one would be bacteria. Bacteria are one-celled tiny organisms. So here's example of bacteria. It's one cell, sometimes it has extensions, and bacteria cause different diseases like tuberculosis, staph, E. coli, strep throat, and so on. Fungi. Here they are. They are plant-like creatures made of several cells usually, and they also can cause diseases like ringworms or yeast infection. Protozoa are protozoa right here, also one-cellular organisms. They can cause malaria, for example. Parasites are living organisms, tiny but complex. They are multicellular, and examples would be roundworms and tapeworms. And we said about proteins. So all of those are called pathogens. And our immune system would be all the cells, tissues, and molecules that mediate resistance to infections, resistance to diseases caused by those pathogens. And when they do it, those cells, tissues, and molecules, when they together fight infection, we call it immune response. Collective and coordinated response to the introduction of foreign substances in an individual mediated by the cells and molecules of the immune system. So our immune system is made of 3 lines of defense, as we call them, or 3 categories of immunity. Barrier defenses, innate or non-specific resistance, and adaptive or specific immunity or resistance. How it works? First, bacteria or virus can get into the body and prevent the entrance that they work for barrier defenses. If those barriers are penetrated and bacteria or virus are inside, causing inflammation, then innate or non-specific defenses start working, killing that infection. But those cannot memorize that response for future. And to recognize that infection for future and prevent that disease from happening for another time, from the second time, remember you never can get chickenpox for the second time. We have adaptive immune response or specific immunity. Now let's talk a little bit more about those three categories. First of all, barrier defenses. They are physical barriers. That is our skin. It prevents entry or remote microbes. No one bacteria and no one virus can get into the body through the healthy and undamaged skin. Our tears would wash away bacteria from the eyes, our saliva, mucous membranes, mucus in our oral and nasal cavities, all of them prevent entry of infection. Next one, if somehow infection is already inside, we have innate or immunity or resistance. It answers fast, it's rapid, but non-specific, so it has no memory for future. And it includes mast cells and basophils, which together provide inflammatory response, which we usually call inflammation. So, number one of innate immunity would be inflammation. Then, natural killer cells, interference, complement system, and phagocytes, different types of them. We'll talk about each of those types of response. Next one, the third one, would be adaptive or specific immunity. It takes time to develop. Sometimes you're already sick, you have that infection caused by a pathogen, but your immune system finally will fight it. Specific immunity will remember it for future. So specificity, ability of our specific immunity to recognize a particular substance, like particular virus or bacteria, for future. Memory, ability to remember previous encounters with that particular substance and respond rapidly. So the second response is faster and stronger than the first, and if we meet the same pathogen again, the disease will not develop. Immune system will kill that pathogen before we get sick. So examples of specific immunity: T lymphocytes, B lymphocytes, antigen presenting cells. How innate and adaptive immune responses cooperate. So right here, that's number one, your physical barriers. Skin, hair, mucus prevent the entrance. Next one, your innate immunity, non-specific part. When pathogens are already inside, right now they are in the throat right here, that inflammation and other parts of non-specific immunity will be killing them. And finally, if disease develops, adaptive or specific immunity will fight it and also memorize it for future. So next time, if we get that bacteria or virus, we will not develop the disease. Okay, number one would be barrier defenses. Let's talk about them. Skin, the surface of the skin, is made of keratinized cells, and those keratinized cells prevent entrance of bacteria and viruses. On the surface of the skin, sweat glands and oil glands or sebaceous glands, they produce their secretions, sweat and oil, that have low pH, and low pH can kill many bacteria, and also washing away action of sweat. In our mouth, in the oral cavity, we have salivary glands producing saliva, which has lysozyme, substance that can kill bacteria. Inside of the stomach of our gastrointestinal tract, we have hydrochloric acid with very low pH, so if we swallow any pathogens, that acid will kill them. Our mucosal surfaces, like mucosa in our nasal cavity, has mucosal epithelium covered with mucus, and mucus also can trap and prevent pathogens from growing on the surfaces. This work also is done by normal flora, special bacteria that are living inside of our body, but they are not causing diseases. Before you continue, please stop the video and answer the questions for part 1.1. We discussed barrier defenses. Now let's talk about innate or non-specific immunity, and we will start from cells. Phagocytes. White blood cells are the most important cellular components of immune system, all types of them. And they are so important because of their ability to move into infected tissues and destroy infection. That is a blood vessel. Those are white blood cells, and they are going into the tissues to destroy bacteria. Now you need to know how they do it. First of all, they should leave the blood vessel and go into the tissues. That process is called diapedesis, changing their shape to exit the blood vessel. You see how they change their shape and they squeeze themselves through the tiny openings between the cells, making the wall of the blood vessel. Usually those would be tiny capillaries. Next one, why would they do that? Why would they go to the tissues? Because they are attracted by inflammatory chemicals produced by damaged tissues. Tissues are damaged by any factor, physical, bacteria. Damaged tissues produce those chemicals. Example would be histamine, and that attracts white blood cells into the tissues. That is called chemotaxis. Main cells would be macrophages and neutrophils, cells capable of phagocytosis. What is the difference between them? Macrophages are large. They are huge cells that can take 1, 2, or more bacteria at a time. How they are formed? They are monocytes. Remember those large blood cells, white blood cells? When monocytes leave blood and enter tissues, they become macrophages. And it takes them a lot of time to do that, but they are long-lived and they can ingest larger particles. They are found in all types of tissues, but more often near spleen, bone marrow, liver, lymph nodes. Next, neutrophils. Neutrophils are always in the blood and they act faster. They are first cells to enter infected tissues. But downside of them, they last only a few hours. So they die, and together with those bacteria that they ingest and destroy, they become that substance that we call pus. So if there is a wound and there is a pus in that wound, that's what it is made of, neutrophils and dead bacteria. So how exactly cells do it? That is a macrophage, large cell. It has cellular cytoplasmic extensions. Those extensions are capable to find and trap stick bacteria, and then extensions pull the bacteria inside into the cell. What happens inside of the cell? The process called phagocytosis. Phagocytosis is endocytosis, or taking something inside of the cell into the cell in vesicles. In this case, that something will be bacteria or viruses. That cell, macrophage or neutrophil, takes bacteria inside in a vesicle, and another vesicle nearby is called a lysosome. It contains digestive enzymes. Two of those vesicles, they merge together, and digestive enzymes start breaking bacteria apart, just breaking them into small particles, killing them. And finally, that bacterial garbage or debris will be released out of the cell. The cell right now can process more bacteria. Next type of cells we are going to talk about would be natural killer cells. Here it is, NK cells. Those are T lymphocytes. And those T lymphocytes, natural killer cells, they can kill tumor, cancer cells, and cells infected by viruses. How they do it? They approach that cancer cell or maybe our own cells infected by viruses and start releasing cytotoxic proteins called perforins. Those perforins are toxic for that cell and they make a hole in the cell membrane. When that happens, a special protein digesting enzyme called granzyme is released from natural killer cell into that cell and causes apoptosis of that cell or cell death. So if it's a cancer cell, it dies. If it's our own cell filled with viruses, it dies too, together with all those viruses, and they cannot spread. Next one, we're done with cells and we go into interferons. Interferons are proteins produced by infected cell. Usually that is our own cell infected by viruses. Here are other viruses inside of that cell. And that cell starts producing proteins called interferons. What do? They go to the neighbor cells and prevent reproducing of that virus in the neighbor cells. How they do it? Viruses, to reproduce, use our nucleus with DNA and RNA. And interference signals the neighbor cells to destroy their RNA. So even if viruses get into that cell, they will not be reproducing. That cell already destroyed its RNA. Next one, what they can do? They can make those neighboring cells to start producing antiviral proteins too. It's not in the picture. They also can cause infected cells to undergo apoptosis or simply to die. When those neighbor cells get infected by viruses, they will die. And they can act not only on the neighbor cells but also on our immune cells like neutrophils, macrophages, and activate them. So they start fighting viruses. Next part of innate immunity would be complement system. It's a group of about 20 proteins that are made in the liver and later can be found in our blood, and they are inactive. They should be activated, and that is a complex series of chemical reactions. It's irreversible. And when it's activated, what do? They bind to the cell membrane of the pathogen that activates it, labeling it for phagocytosis. So other phagocytic cells can recognize the pathogen and destroy it. That process is called opsonization. Next one, those complement system proteins can diffuse away, move away from the pathogen and act as chemotactic agents to attract phagocytic cells to the site of inflammation. Finally, they can form damaging pores in the plasma membrane of the pathogen, killing it. Okay, let's take a look at this picture. That's an invading pathogen, let's say bacteria, and proteins, those complement system proteins, bind it, so right now immune system cells could recognize it and destroy. They also, those proteins, can attract phagocytes to the infection site, so phagocytes would arrive and destroy that pathogen. Finally, when they are activated through that long chain of chemical reaction, they can produce membrane attack complex. That green one is a pathogen. Membrane attack complex, when those proteins are making a hole in the pathogen's plasma membrane. Water rushes into their cytoplasm, causing that pathogen to die. One more type of innate immunity is inflammatory response, or simply inflammation. So that is a tissue that was injured, maybe by a splinter or cut with knife or whatever it is, and some bacteria got inside, pathogens. So the damaged tissues immediately start producing chemicals, mostly histamine, and histamine, that's what is causing the inflammatory response. So tissue injury, regardless of type of injury, can cause inflammation. And response is initiated by chemical mediators, histamine. So what happens? First of all, vasodilation. Blood vessels become wider and more permeable. So white blood cells can go from the blood to the tissues to kill those pathogens. Remember, chemotaxis and diabetes. Now, when blood vessels become more permeable and dilated, that causes redness right here, because not only white blood cells, but some amount of blood plasma goes right here into the tissues, causing swelling. So swelling, redness, warm because of such amount of blood in this area, and of course pain because of the injury. Recruitment of phagocytes, all phagocytic cells, we were talking about them, macrophages, neutrophils, they go into the tissues. That is inflammation. Now, there are two main types of inflammation: local, specific area of the body, like a splinter in a toe, for example. And local symptoms would be redness, heat, swelling, pain, and loss of function, because it's difficult to operate with something that is in pain and swollen. Next one, systemic inflammation. Systemic inflammation, when that process of inflammation happens in many parts of the body. Same symptoms as local, but also in addition, there will be increase in neutrophil numbers. So if you take a blood test, there will be high level of neutrophils in the blood. Fever. Different types of cells produce substances called pyrogens, and they cause increase in body temperature, fever, and that is important for performance of immune system. And also some types of bacteria cannot reproduce at higher temperature. And finally, that widespread increased vascular permeability due to histamines causes large volume of blood plasma to leave blood vessels and enter interstitial spaces. And in some cases, it can cause dropping blood pressure drops significantly, and it can lead to shock. Before you continue, please stop the video and answer the questions for part 1, part 2. Now we will talk about the adaptive or specific immunity. Why it is called specific? Because it involves the ability to recognize, respond to, and remember a particular stimulant. So, repeated exposure would lead to faster and stronger immune system response. If a child had mumps, measles, or chickenpox, that kid's immune system remembered a virus or bacteria that causes a disease, virus in that case, and in future, if that kid who already recovered long time ago from that chickenpox, if the kid is exposed to the same virus again, the immune system would respond much faster and stronger, and the disease will not start again. You never can have that type of disease again. Please pay attention that starting from this slide, we will be only discussing the adaptive and specific immunity. We're done with barriers and we're done with non-specific immunity. So what can be those stimulants that cause a response from specific immunity? Antigens. You remember that antigens are large molecules, proteins or glycoproteins that are located on the surface of cells or viruses, bacteria that trigger an immune response. Most of the antigens are foreign. They are not produced by body. They are introduced from outside. First example would be pathogens, bacteria, viruses, and all those other types of pathogens we listed earlier. They cause disease. But sometimes our immune system thinks that tiny particles like pollen, animal dander, feces of mice, some types of foods or drugs, our immune system thinks those are antigens and it overreacts. It tries to kill them, and that is called allergic reaction. So the immune system's first exposure to a pathogen, when a kid with a chicken pox gets sick for the first time, that is called a primary adaptive response. And the symptoms of the first infection that kid has, the fever, all those skin lesions, it's called primary disease. And those symptoms are always relatively severe. Later, the secondary adaptive response is stronger and faster. It can happen years later, and in this case, immune system will answer so fast that pathogen, virus, will be eliminated from the body before it can cause significant tissue damage or any symptoms. So that secondary response is the basis of immunological memory, which protects us from getting diseases repeatedly from the same pathogen. Finally, there are also self-antigens. They are produced by body and our immune system reacts to them. They are used as markers to allow adaptive immune response to differentiate self from non-self, self-recognition. And usually, our immune system is not supposed to respond to self-antigens. But it still can happen. If it responds, there are two variants. First, response to self-tumor antigens. When our own cells start growing and turn into tumors, cancers, they change so much that our immune system cannot recognize them as self anymore and starts attacking them, which is very good and beneficial for us. But sometimes our immune system makes mistakes and starts thinking that our self-antigens located on our own cells and tissues, that those are pathogens, and starts destructing those systems or those tissues. That is called autoimmune diseases. Adaptive or specific immunity operates by two mechanisms. One is called antibody-mediated or humoral immune response. It's provided by B lymphocytes, and B lymphocytes produce antibodies that work between the cells in extracellular fluids, eliminating bacteria, viruses, and their toxins. Those antigens produced by B cells, they clump antibodies produced by B cells, they recognize pathogens, clump them together, and destroy them. Second type of immune response would be cell-mediated, and it's provided by T lymphocytes to eliminate intracellular pathogens. So when our own cells are already invaded by bacteria or viruses, then T cells can recognize it and form special activated killer T cells that will kill infected cells, and also phagocytes if there are pathogens inside of them. The chart shows you excuse me, main differences between antibody-mediated and cell-mediated immunity. Before we even start going into details of those two types of immunity in part 2 of this lecture, you need to know main differences. And here they are. Antibody-mediated immunity is provided by B lymphocytes or B cells that originate from red bone marrow, and they mature in red bone marrow also. Mature cells are located either in blood or lymphatic tissues, and primary product fighting infection would be antibodies. They protect us against extracellular antigens like bacteria, toxins, parasites, and viruses outside of our cells, in the fluids between the cells. Second type, cell-mediated immunity, is provided by T cells, which are born in red bone marrow, but then carried to thymus for maturation, and they are also located in blood and lymphatic tissues, and primary products would be chemicals called cytokines. They protect us, T cells and their products protect us against intracellular antigens, viruses, bacteria, and fungi located inside of the cells. Also, they protect us against tumors. Okay, so those are main differences that you're supposed to know before you start the second part of this chapter 21. Please remember to answer the questions for part 1.3.